Hello, and welcome to the next Bitesize Bioinformatics talk. I am Stephen Wingett, and I'm going to be talking to you today about contamination detection with FastQ Screen and Kraken. This talk is going to be just a little bit different from most of the ones we've had previously, and that's because I'm having to pre-record it and then upload it, and then screen it as live via Zoom. Unfortunately, I'm having to work from home at the moment, and my internet connection is a little bit slow. That's why I'm having to do this. But all being well, when the talk is finished, I might be able to answer any questions you have via Zoom as per usual. Okay, just a couple of notes on Zoom. When you're actually viewing the talk, if you move the mouse pointer to the bottom of the screen, a menu bar should appear. From here, you should be able to send a direct message or a chat to the group using the chat button. Or alternatively, if you want to get my attention, if you just raise your hand, then hopefully at the end of the talk I'll be able to discuss the point with you want to discuss with me. Okay, so without further ado, let's get on to the talk about FastQ Screen and Kraken. So it is around a decade ago now that the first next generation sequences became available. They changed forever the field of molecular biology and generated far more data than had previously been seen in the field of sequencing. This new technology created a great demand for new computing techniques, and a range of tools were developed at the time for the management, analysis, and alignment of data generated on next generation sequences. Around about this time, we at the Babraham Institute developed the tool FastQ Screen. FastQ Screen allowed you to quickly assess the quality of your data. Specifically, this tool was designed so that the user could quickly confirm that the reads in their sequencing run were derived from the organism that they would expect. This might sound at first glance a sort of trivial question to address and not necessary, but actually there are lots of reasons why maybe the reads in your sequencing run are not what you expect. Superficially, I guess, there could be some sort of sample swap. Also, experiments that involve amplifying up a small amount of starting material are particularly prone to amplifying up the wrong molecule. So you could have a sample with a small amount of contamination from a different source, and then you amplify up the wrong type of molecules. Primarily, FastQ Screen is designed to identify interspecies contamination. FastQ Screen does this by using one of the three aligners, Bowtie, Bowtie 2, or BWA. FastQ Screen then uses these aligners to map your sample of interest to a range of target genomes. These should be the genome that you expect to see in your sample and a whole range of possible contaminants. Such contaminants would either be adapters used in the sequencing or maybe some sort of vector molecules used in the library construction, or perhaps you could use other species used in your lab. For example, if you are working with human samples and somebody else in your lab is working with yeast samples, it would be a good idea to use FastQ Screen to check that no yeast DNA is present in your datasets. Over the years, we have found FastQ Screen to be an invaluable tool to check that samples do indeed contain what they're supposed to. Also, the tool has been invaluable in identifying samples that are contaminated or not containing what they should do in some other way. For these reasons, we have integrated FastQ Screen into many of our bioinformatics pipelines, and in so doing that samples which come off our in-house sequencer are routinely screened by FastQ Screen. Okay, so now I'm going to discuss some of the results that could typically be generated by FastQ Screen. So FastQ Screen takes as input FastQ files. It then analyzes these, maps them against the target reference genomes using one of the pre-specified aligners, and then generates results files. Results files include a text format file listing the number of raw reads that map to each genome. It also summarizes this in a graphical format, which is what is shown below. There is also an HTML output file, which includes a graph of the results and the full numerical results listed in a table. In this example here, we should be working with a mouse sample. Having analysed the data with FastQ Screen, the data do indeed suggest that we have mouse DNA. If you look at the graph, you can see along the x-axis all the genomes to which the reads were mapped. So that starts with human, mouse, rat, and then there are a range of other genomes which you might not be able to see the writing too clearly, but they are E. coli, Drosophila, and so on. And right at the end, we have no hits. So what does this mean? Well, each bar represents the percentage of reads that map to each genome. So if you look at mouse, which is the most abundant genome, we see around about 80% of the reads map to mouse in some way. Around 15% of the reads map to rat, and maybe around about 5% of the reads map to human. You will also see at around about the 20% mark no hits. These are reads which map to no genome whatsoever. What you may have noticed from this graph is that the total does not add up to 100%. That is because any given read may map to multiple genomes. For example, a read may map to the mouse genome and the rat genome. Reads that only map to one genome are shaded in blue, be that light blue or dark blue. Conversely, reads that map to multiple genomes, that is 2 or more, are shaded in either dark red or light red. What you can see in these results, therefore, is that by the largest category are reads which map to the mouse genome and only the mouse genome, which strongly suggests that this is in fact mouse DNA. While it is true that a small proportion of the reads that map to the mouse genome do seem to appear to map to rat or human, this may be as expected due to the close evolutionary similarity of these species. Interestingly, these reads that multi-map to the mouse and other mammalian genomes do not appear to map to other species, such as Arabidopsis or Drosophila and so on. So, these results are strongly suggestive that we have indeed sequenced mouse DNA and everything looks as it should. One thing I just would like to point out as well is the distinction between the light and the dark shaded red and blue colours. Light blue means that we are looking at reads that map, while only to one genome, they also map uniquely to that genome. Whereas, in contrast, dark blue reads will only map to one genome, but could map multiple times to that genome. So, for example, if you look at the dark blue bar in the mouse column, these are the number of reads which map only to the mouse genome, but will have multi-mapped to the mouse genome. The light red reads are those which, while mapping to multiple genomes, only map uniquely to the genome shown in the bar. So, for example, if you look at the rat bar, the light red shading reflects the number of reads which map to rat and at least one other genome, but when they map to the rat genome, they only map to one specific location. In contrast, the dark red shading reflects the reads which map to rat and at least one other genome, and when they mapped to the rat genome, they map to multiple locations. Okay, so we've seen a good sample, so what might a bad sample look like? Well, I'm going to give you a series of different data sets now that we encountered over the years, and I'm just going to talk through with you the type of problem we're seeing and you know, what type of artifact we're looking at and how it may have been generated. So in this first example, if you look below, we have a similar graph to how we did before. And what we should be seeing is mouse DNA. So we can see that we've mapped the reads against all the different genomes as before. And yes, the reads do map to mouse as before. There are reads which map to mouse and only mouse, which are in the blue colours. However, if you look at the bar next to it, the human reads, you'll see there are a sizable number of reads, in fact more reads, that map exclusively to the human genome as compared to mouse. This suggests there is human DNA in the sample, and that the sample has been contaminated. Not knowing the source of the DNA is a problem for a number of reasons. But when it comes to the analysis, when you align the reads, for example, against the reference genome, what you may find, however, is that the human contamination is then mapping to the mouse reference genome. So what you have now, you've incorporated errors into your analysis. This may be particularly problematic if, for example, the human sample was ChIP-seq, which has clearly defined peaks. However, by using FastQ Screen, you have been able to QC this dataset, and most likely you would want to remove it from further analysis. So let's now look at a similar example. This dataset was supposedly a single-cell mouse DNA. In this example, I've truncated the graph a little bit, just so you can see the bars which are of interest. As you would hope to expect, there are indeed a sizable number of reads which map to the mouse and only the mouse genome. However, what you can see is there are a large proportion of reads which map exclusively to E. coli, suggesting that we have E. coli contamination in our sample. This will be bad enough on its own, but if you look at the graph to the right, what you can see is the degree of E. coli contamination in each single cell sample. What you'll see is that some of the samples only contain a small amount of contamination, while some are almost exclusively E. coli DNA. What this means is when you're looking at comparisons of different cells and you observe differences, what these differences are may not in fact be biologically important, but just may reflect the degree of contamination of E. coli DNA in each of the different single cell samples. So this is an interesting example that we downloaded from a public repository. We downloaded the data and then performed FastQ screen analysis on it, and we had a result like this. And it's supposed to be mouse DNA, and that's what we see. It's got pretty good mapping efficiency against the mouse genome. About 80% of it maps, and only 20% doesn't map. And that all looks well and good. But then what's suspicious is that, in fact, none of the reads seem to map to any of the other genomes, which is surprising just because, we do expect to see some degree of sequence identity between genes in different genomes. Also, what's interesting is that the reads that map to the mouse genome map uniquely to the mouse genome. They only map in one specific location. This is very unusual. In fact, you really shouldn't expect to see this in any normal data set. What we think has happened is that the data submitted to the Genome Archive as a FastQ file has actually been filtered, and all the multi-mapping reads have been removed before it was submitted. We're not completely sure of this, but this seems to be the most probable explanation for this data set on cursory inspection. Obviously, this is not what you would expect to see, and of course, if you wanted to do your own analysis on the data, you have to now bear in mind that it may have been pre-filtered or manipulated in some way, which of course, in which point you start to lose confidence that the conclusions you reach are actually valid. So here we have a slightly more complicated example of a potential problem. So what we did to get hold of the data, we went once more to the public repositories and we downloaded a large number of mouse RNA-seq datasets from the repositories. Because we were trying to download a huge number of datasets, this wouldn't actually be practicable in terms of the data download and the amount of storage required on our systems. So what we did is we downloaded the first several 100,000 reads from each dataset and then performed the FastQ screen analysis on that. Now what you might hope to expect is the first several 100,000 reads would be fairly indicative of the whole dataset, but here that proved not to be the case. So if you look at the top graph, what you see is the results based on the first 100,000 reads or so. If you look at the mouse bar, what you can see is there is around 10% of the reads or so map to the mouse genome and to no other genome. However, there are a large number of reads which map to multiple genomes, and what that appears to be is our mitochondrial dataset. So we have a number of mitochondrial genes in our test genome, and these are derived from a number of species, including mouse. And what you can see is that these reads in our sample appear to map to mouse and to our mitochondrial genome. Now that all sounds fairly reasonable. It would just be that for this particular data set, we see a high degree of mitochondrial contamination. However, as we successively downloaded more data from this sample, what we saw was the relative proportion of mitochondrial reads decreased. So if you look at the second bar, again, we can see a similar sort of pattern, but here the proportion of reads which map to the mouse and only the mouse genome has increased, i.e. those in the blue shading, whereas the number of reads which map exclusively to the mitochondrial genome has decreased. So how would we explain this? Well, I think the best way to explain this is that the raw FastQ file had been sorted in some way prior to uploading. Obviously, this doesn't mean that the FastQ file is invalid, but it just would cause us to raise a few eyebrows and make us wonder whether the data had indeed been manipulated before it had been submitted to a public archive. If I were working on these data sets and trying to draw conclusions from them, I would bear this in mind, and I would probably be a little bit more cautious about reaching any valid conclusions from this particular data set. So in this final example again, we have a public RNA-seq mouse dataset, and we've run it once more through FastQ Screen. What you see once more is that a large proportion of the reads map to the mouse genome, which is as should be expected. However, what we also see is a large number of the reads map to many other of the genomes, such as closely related species, such as human and rat, but also, in many ways more worryingly, to more distantly related species, such as Arabidopsis or yeast. We really wouldn't expect to see this from a valid RNA-seq mouse dataset. Also, what's also more worrying is that many of the reads map to multiple genomes and to multiple locations, i.e. all the reads that we can see here are represented by the dark red shading. I would interpret this in that the data contains low complexity sequence, that is, very simple repeat sequences, which by virtue of their low complexity can map to multiple genomes. On the basis of this, I would most likely want to discard this sample from my analysis. But this underlies the importance of valid QC, for if I didn't perform QC on this dataset and just double check everything was looking as it should, I may just naively assume that around about a 90% mapping efficiency had been achieved and this was a good sample. Whereas in reality, what we seem to have done is sequence some low complexity molecular artifact. At this point, I'd like to briefly mention Millie Thackeray, who was a placement student working at the Babraham Institute, who did this work. So she was the person who was responsible for downloading a large number of mouse RNA-seq datasets and then analysing them with FastQ Screen. We all found it quite a surprise, actually, the number of publicly available datasets, which on closer examination are not quite what you would expect. So let's suppose you're interested in using FastQ Screen on your own datasets. How would you go about getting hold of it? Well, the homepage for the tool is listed below. This is the Babraham Bioinformatics website /project/FastQ Screen. So if you just type this link into a web browser's URL bar, the home page for the tool should pop up. Alternatively, you may want to navigate to the project's homepage via the Babraham Bioinformatics website, which is shown here. So if you want to go to the project's homepage, you just click on Projects, and then what you will see is a list of all the projects that we've either developed or are currently developing in the Babraham Bioinformatics group. We have other tools quite well known there, such as FastQ, Hiccup, Labrador, and so on. But FastQ Screen is also here, and if you just click on the link, you'll go to the FastQ Screen homepage. So what you will see here is just a brief overview describing what FastQ Screen does. It tells you the programming language that it uses, which in this case is Perl, a list of the requirements and dependencies for the software, and other things such as the code maturity, license, and so on. And then we have a brief description of the software, sort of overview of the output you get, and the changes that have been made to the software over time. Also from this page are a series of useful links. So if you want more information about the software and how it works, how to run it and how to install it, etc., you just click on the online documentation link, and that will take you to this page. So this essentially constitutes the manual for the tool, and everything you would want to know to run the software hopefully should be included in these pages. So they're quite extensive pages, and they cover a number of topics which are shown on the shortcut links bar on the left hand side, and it tells you sort of how to download the software, more details on the requirements, takes you through the installation process, more details on interpreting results, and so on. So hopefully everything you should need should be in these read the docs format instructions, which is fully searchable using this search box here. I should just point out there are a series of FastQ Screen online tutorials that I've already prepared. So I've talked quite generally about the tool here, but these videos provide a more detailed sort of technical information on setting up the software and running it. So if you're having any problems and you can't quite understand the documentation, then this is not a bad place to go to get more information. What you will also see in the documentation is a link to GitHub. So this is where we host and store the code for the software, and we keep a track of all the previous versions that have been released. So if you want to download a specific older version of FastQ Screen, you can go here. This is also the way you can report any bugs regarding the software as well. So if we go back to the home page now, if we want to actually download the software, we would click on the Download Now page. And here we have a list of options for FastQ Screen. So this would take us back to the home page. This would be a README giving us general notes. We have release notes. But here we have the link to download the software. So if we just click on that, we will download the scripts, which are actually just quite a series of quite small Perl scripts. They're bundled up in a tar archive, so if you just unzip those, and then you should be able to run them on your system. Just incidentally, this FastQ Screen is geared for Linux operating systems. Also worth knowing is if you want to test FastQ Screen to see if it works, what you can do is download a test dataset, which comprises a small number of reads derived from a human sample. So essentially you've got a very small FastQ file. You can download that, process that with FastQ Screen, and then look at the results. I've also included the results in there that you should expect to see if FastQ Screen has run correctly. So if you run the software on your system and then check that the results you get match what I believe you should get, If those two correlate very well, maybe not exactly because you can just get some variation due to different versions of aligners or what have you, but so long as the results pretty closely agree, you should have confidence that FastQ Screen is working on your system. So I'm very briefly going to talk about running FastQ Screen. I'm not going to talk about this in any detail just because, as I mentioned, there are tutorial videos already available uploaded to YouTube where you can get more information on this. But just to summarize, FastQ Screen works on a Linux system via the command line. So I have already installed FastQ Screen on this system and it is added to the system's path. So if I type in FastQ Screen, we'll see the help message displayed for the software. Okay, so if we just look, we have already downloaded the test dataset, which I spoke about a little bit earlier, and we're going to run FastQ Screen on this. So we've set everything up, we've installed it, we've got a configuration file ready to go, we've got the aligner Bowtie 2 already available, and the genome indices have been put together. So all we would need to do is to run FastQ Screen and specify the test data set that we want to analyze, which is that, and then we press enter and off it goes. And what you'll see initially is it's going to add all the databases that I've specified, and it's going to blast against all of these. So as you can see on our cluster setup here, we check reads against a large variety of different genomes: human, mouse, other mammals, then we have plants, potential artifacts, viruses, that sort of thing. So what it first does when it runs is it will subset the data. So for QC analysis, we don't actually have to map everything in the data set. We just want to map a small proportion of the data to get the results that we would expect. Now, it's possible if you just analyze a small subset of the data at the very start of the file, this may not be indicative of the dataset as a whole. The first few thousand reads or whatever may just be actually just ends. These are essentially reads in which the base couldn't be called by the sequencing software. So what we do, well, what FastQ Screen does, it will subset the data set. That means it will go through the data set, and every 1,000 or so reads, it will extract a read and make a small subset data set. And then it's this that it uses to blast against the different genomes that we've selected. So what we can see here that we've added our genomes, we have performed the subset, and now we are mapping our subset of reads against all of these different genomes. And this is going to take a little bit of time to run. So what I'm going to do is I'm going to pause the video, and then when this is done, I'm going to show you the final results. Okay, so the processing has now finished, and what we can do is if we list the output, we can see the original file still there, so that shouldn't be altered, and we see three output files. There is a PNG file, and that is just an image of the graphs showing the results. So these are the graphs that I've been showing you previously in this online seminar. We also see the text file of the results, and we see an HTML file, which can be viewed in a browser such as Chrome or Firefox, and that shows an interactive graph of the mapping results, similar to the PNG file, and it also shows the raw number of reads mapping to each genome, as we see in the text file. So if we just wanted to look at the contents of the text file, we could just use the cat command and have a look. And there we go. So we just have a nice tab delimited set of results of all the reads mapping to each genome and the relative percentages of those. Okay, so that's the output. That's what you should see if it's worked. But if, as I said, if you want more information on this or more technical sort of overview of the setup and running, then please refer to the other YouTube videos that I've already prepared. So one of the extra features of FastQ Screen is that it is compatible with Bismarck. So if you don't know, Bismarck was a tool developed at the Babraham Institute primarily by Felix Kruger and is used for mapping bisulfite sequence data. So essentially, FastQ Screen uses Bismarck to map the data, and this will allow you to perform interspecies QC on bisulfite libraries. It is also worth mentioning at this point that FastQ Screen is compatible with the tool MultiQC. So MultiQC was developed by Phil Yules, who was formerly at the Babraham Institute. What this tool does is allow you to aggregate lots of different forms of analyses from bioinformatics tools and pipelines into a consolidated single report. A FastQ Screen module was incorporated into MultiQC from the beginning, and as such provides an excellent way to compare a large number of samples. So let me show you an example. So here is an example output from MultiQC, and here specifically we're looking at the FastQ Screen component. What we can see are all the different samples stacked on top of one another. And we have horizontal bars representing the number of reads mapping to each type of the genome. So what we can see here is that the majority of reads for each of the different samples mapped to the light blue genome, which in this case corresponds to human, as you can see here. Now, if that's not immediately obvious to what's happened, if you hover over any of the bars, you can see the key in more detail with the species name highlighted and the number of reads. So what we can see here is around 85% of the reads mapped to the human genome and 15% multi-mapped to more than one genome. And that's a nice way. And this is a very nice way to simply compare the data. So if you do have a large number of data sets which you want to analyze with FastQ Screen, I would certainly recommend running MultiQC on your FastQ Screen output after FastQ Screen is finished. This provides an excellent way to quickly and simplify any samples in which there may be a potential problem. So far we have been talking about FastQ Screen as primarily a QC tool. That is to judge the quality of FastQ files that you pass to it. However, with time, we developed some extra functionality to the tool. That specifically is the ability to filter FastQ files. So in this simple example, we have a FastQ file in which we have reads which come from two separate genomes, genome A and genome B. And the idea would be that we would pass this FastQ file to FastQ Screen. It will then map against the genomes which we're interested in and identify the reads which map unambiguously to genome A. That is, reads that map to genome A but do not map to genome B. This way we can filter FastQ files to reads that map to one genome and filter out some kind of background genome. This has been found to be useful in the sequencing of enteric bacteria. The idea would be that the bacterial sequences are sequenced and then the host organism's genetic background could be removed from the dataset. As I mentioned before, it is a FastQ file that is passed to FastQ Screen during this filtering, and it is a FastQ file that is returned post-filtering. Therefore, you have a new file which you can go on to process as and how you need it. For example, you may want to pass it to a standard aligner to perform some sort of mapping. The options for the filtering are really quite versatile, and so you can set up the filtering in a number of ways. For example, you may want to extract the reads that map to none of the selected genomes. You then have a FastQ file which you can interrogate further to try and find out the source of these unknown reads. Or we have encountered people using the reads to filter for reads which map uniquely to a genome of interest, i.e. they are not multi-mapping reads, that is, reads that map to multiple locations within the same genome. So that's a brief note on the filtering of FastQ files. It is interesting how a tool that was originally designed as a bit of QC software can then go on to perform analysis and filtering in its own right. I just wanted to talk briefly about a couple of interesting asides regarding FastQ Screen. When you write a bit of software, it can be quite interesting to see how other people are using it and to the diverse range of applications to which it can be put. I'm going to discuss two of the most notable of these. Firstly, a team at York University in the UK used FastQ Screen in work involving 13th century pocket Bibles. Pocket Bibles were different from their predecessors in that they had very thin pages, which meant that they could be put in a pocket. Following their introduction, the numbers of them boomed in the 13th century, and there has been much speculation on how this new technology came about. A couple of ideas are that some sort of new rendering process made it possible to make these ultra-thin pages, or another possibility is that some new animal hide was used in the construction of the parchment. The team went out to investigate the sources of the vellum used in these Bibles. Foremost, they used a mass spectrometry technique to do this. However, they also devised a technique where they extracted ancient DNA from the vellum, sequenced it, and then were able to process it with FastQ Screen. By the end of their analyses, they concluded that there were no novel and unusual animals' hides used in the construction of the vellum. As I mentioned, certain animals such as squirrels were put forward as possible candidates for the reason it was suddenly possible to generate these ultra-thin pages. Essentially, the hides seemed to be restricted to the usual cattle such as cow, goat, and sheep. Interestingly, the type of animal hide used in the construction of the Bible matched very well with known distribution of those particular animals in the 13th century, further lending support to the idea that the vellum was derived from these species. They concluded, therefore, that the use of new species did not seem to be necessary for this innovation, and rather they suspected it was due to some new sort of technical innovation. In my second interesting example of how FastQ Screen has been used outside the Institute, I wanted to talk about the case of Ötzi the Iceman. Now, Ötzi was discovered in 1991 in the Tyrolean Alps by a group of tourists. When they first discovered his body, they believed, due to the fact he was actually relatively very well preserved, that he was a recently deceased mountaineer. However, on further examination, it was realized he was a naturally preserved mummy from around the year 3400 BC, which is known as the Copper Age. He was named Ötzi, who is now actually Europe's oldest known natural human mummy. The mummy was obviously of much valuable anthropological interest, and a number of scientific studies were conducted on him, one such of which was to identify the origin of his animal clothing. Owing to the mummy being well preserved, it was possible to extract ancient DNA from the sample, which could then be sequenced and passed to FastQ Screen to identify the source of his apparel. Following this, it was possible to identify that his hat was originally derived from the fur of a bear. The quiver in which he held his arrows was derived from the hide of a roe deer, and his loincloth was made from the hide of a sheep. Needless to say, I found this very interesting and quite delightful that a tool we had put together at Babraham to QC the samples that had come off our sequencer had been used in such an unexpected and fascinating way. If you would like to read more about FastQ Screen, in addition to the online documentation, I should just let you know that the software has been published in the online open access peer-reviewed journal F1000. So far I've been talking about FastQ Screen and how in most circumstances you can probably predict what the source of contamination is going to be in your sample. So that could be something else you're currently working on in the lab, or maybe some organism somebody else is working on in your lab, maybe some sort of bacterial species you've been using to create your library, or something else that's been run on the sequencer. However, whatever it is, you can probably have a reasonable guess at the source of likely contamination. And that's all well and good in most instances. However, there are situations when you'll get contamination in a sample and you won't know the source in advance. And for these situations, FastQ Screen simply won't identify it. This is because you won't have included your contamination species in your list of target genomes. FastQ Screen therefore won't search against that, and therefore all the reads which are the result of the contamination will be assigned to the no-hits category in FastQ Screen. So, is there anything you can do about this? Well, yes there is. There is a tool called Kraken, which was developed by a team at Johns Hopkins, which was primarily aimed at metagenomic studies. So these are the experiments where you would take a sample of some kind, and within that sample you would expect to find a large number of bacterial, archaebacterial, or viral species. So you could then go on to sequence these samples and then pass them to Kraken. Kraken uses a pre-built, exhaustive large database, including a vast array of different species, and then uses an algorithm to assign the reads to each of these different species. If reads cannot be assigned to a specific species, Kraken will try to assign them to the phylum or the genus or the order as appropriate. So Kraken uses a KEMA-based alignment system, and I'm going to talk about that a little bit later. So although Kraken was originally designed as a metagenomics tool, it still provides an excellent tool to identify contamination from a range of bacterial, archaea, or viral sequences. So I just wanted to talk briefly about how Kraken performs these assignments. So if you look at the diagram on the screen, on the top part of the figure, you will see your query sequence. This is the read that you're trying to classify. So what Kraken does is it subdivides this read into a series of k-mers. I believe they're 31-mers to be more accurate. And then what it does is it will assign these k-mers to a particular species in its reference genome. If it can't assign to the species, it will then work its way up along the taxonomic tree. But Kraken is trying to assign as far down the taxonomic tree as possible. So each of the different k-mers have been assigned to different parts of the taxonomic tree. That's all well and good, but now we need to make a decision about where exactly we're going to allocate this specific read. So what Kraken then does is it prunes the main taxonomic tree to create a subtree, which contains all of the nodes to which a k-mer had been assigned. There is also a scoring system here, which refers to the number of k-mers which were assigned to each node. So if you see the figure 1, number 1, that means that 1 k-mer was assigned to the node which was shaded in purple, 10 for light blue, 4 for orange, and so on. Kraken will then try and work out, working back through the pruned subtree, the node which will give the highest score, which in this case is the orange node. So what that means is if you work your way back from the leaf to the root of the tree, you'll get a score of 4 plus 10 plus 1, which is 15. And this is the highest score possible. And then Kraken goes on to assign the read to that particular taxonomic classification. So this is how Kraken assigns reads to taxonomic classifications. It will try and assign directly to species, but if that's not possible, it will work its way up the taxonomic tree. So I wanted to talk about a case study using Kraken that we had, and how it's used for contamination detection. So we received an email some time ago from somebody who had been using one of these test at home DNA kits. So the idea is you provide a sample of your own DNA, you then send it off, and the company will give you information based on the sample that you send. Also, as part of the deal, they were offering to supply the FastQ file associated with that particular DNA sample. So the customer was quite interested in being able to study their own sequences and had the bioinformatics sort of capabilities to do this. So they got the FastQ file back and they started analyzing it. And what they were concerned was that a large number of the reads in the FastQ file couldn't be assigned to the human reference genome. And as it turns out, there have been quite a few people with this company who've been having similar problems. So if you look at the figure here, what you can see, actually this one isn't too bad, but around about 60% of the reads do map to the human genome, which is good, but then there's a substantial number of reads which don't. For the particularly poor samples, the number of reads which map to the human genome was very low, and I think we were talking around about under 20%. So the people wanted to try and so the person who got in contact with us really wanted to understand what was going on, and they wanted to understand why they couldn't map their reads to the genome. So we ran it through FastQ Screen, as you can see, and we validated that they were in fact getting poor results. So then we dug a little bit deeper, and we used FastQ Screen. We used the filtering options to extract all the FastQ reads which didn't map to the human genome. We then took this FastQ file and went on to analyze with Kraken. So what you can see here are the first few results that we got back from Kraken when we used it to process these reads which didn't map to the human genome. And this is the sort of standard output you get from Kraken. So right at the top here, what we have are the proportion of our reads which were unclassified by Kraken. So it's around about 58% were unclassified. However, Kraken was able to classify a sizable amount of them, around about 42%. So that's the 42% here, and they were able to assign the root of the tree, i.e. that's all organisms within the Kraken database. And of these, around about all of them were able to be assigned to cellular organisms. And likewise, around about all of them were able to be assigned to bacteria. So around about 42% of these reads. So as you can see here, what we have here is the establishment of a taxonomic tree as this indents to the right. So D means domain of life, and then we have phylum, class, order, family, genus, species. Just to explain the taxonomic, just to explain this output a little bit more, as I mentioned, this is the percentages, and this is the taxonomic classifier, a level classifier. This is the taxonomic ID of each of these species or family name or whatever. So the taxonomic tree of Neisseria is 482. So we have two columns of reads here. These refer to reads which were assigned at a particular level, and these refer to reads which were assigned directly at that particular level. So that's probably a little bit confusing. So if we just look here, what we're seeing here is bacteria. Okay, so we have a domain here, bacteria. The 399 means that 399 reads were assigned directly at the level bacteria. They couldn't be assigned at any lower down the taxonomic tree. They could only be assigned just to bacteria. Whereas this number here reflects the number of reads which fell within bacteria at any of the subsequent levels. So did these results we got back from Kraken make sense? Well, we believe they do. As I mentioned before, we had a large proportion of Neisseria reads, and we had a large number of reads which mapped to Prevotella species. And we had a little search on these a little bit more, and these are common bacteria found in the mouth. Now, what was interesting is when we looked at the protocol for obtaining the DNA from the human participants, is that a mouth swab was used. So you'd place this in the mouth, rub it against the side of your cheek, and then you'll be able to collect cells from the side of your cheek. But no doubt, while doing this, you also transferred a large number of oral bacteria onto the swab. So these bacteria now would be on the swab, they would go through the library prep and be sequenced. So we believe that what's actually happening with these home testing kits is we're getting a large number of reads back, but a significant proportion of those are actually just sequencing the metagenome of the mouth, which you may argue is interesting in some ways, but was not exactly what the person who performed the home testing kit was actually interested in. So to summarise, FastQ Screen was a tool developed in the Bioinformatics department at the Babraham Institute, and it's used to QC next generation sequencing data. And it's a very quick and easy way to make sure that the reads you're getting out of the sequencer do indeed map to your expected genome. So if your reads aren't mapping to your expected reference genome, what you should also pass to FastQ Screen are a list of genomes which are likely sources of contamination. FastQ Screen will then map your reads against these likely sources of contamination and tell you, indeed, if there is contamination and what the likely source is. Using FastQ Screen is also a good way to check for other potential problems and artifacts which may have occurred during sequencing or the library prep stage. For example, running the software is a good way to identify datasets with a high proportion of low complexity reads. FastQ Screen itself is written in Perl, and it uses either Bowtie, Bowtie 2, or BWA as the reader liner. As I mentioned before, it's compatible with other tools such as Bismarck, so you can perform QC on bisulfite datasets. And if you're analysing a large number of datasets in one go, then FastQ Screen is compatible with MultiQC, which is a good way to collate the results from large numbers of samples in a nice, simple and easy to read output format. We use FastQ Screen widely at our institute, and we know it's being used outside the institute as well. If you don't know the source of your contamination and really can't guess at it, then we recommend using Kraken as a way of identifying the source of contamination. Kraken is a metagenomics tool and contains an exhaustive database of bacterial, viral, and RKA sequences. Using Kraken in conjunction with FastQ Screen should help you identify, hopefully, most sources of contamination in your datasets. So thank you for listening to my talk on FastQ Screen and Kraken and its use in contamination detection. That comes to the end, so thanks for listening. I'm now going to endeavour to try and answer any questions you may have via Zoom. So hopefully my internet connection is stable at the moment and the network's strong because I'm having to do this through my mobile phone. So yeah, thanks for listening and goodbye.